Cassidy-Driscoll is going to take this stage and introduce our next speaker.
So it is with great pleasure that I introduce our next panel, which is the Optogenetics panel. We will have three very talented doctors. Dr. Raj Agrawal is an ophthalmologist and retinal specialist and is currently the Vice President of Clinical Development and Therapeutic Area Head at Therapeutics. Dr. Agrawal is also the founder and president of Radisson Global, a nonprofit organization that provides sustainable solutions to retinal disease management around the world. He is also the founder of the Orange County Eye Project that manages the eye clinic providing specialized eye care to patients in Southern California. Previously a clinical faculty at Donny Eye Institute and University of Southern California. He is the co-inventor of the Argus 2 device, the first ever treatment approved for patients with advanced retina pigmentosa.
Good morning again. Just to remind, my name is Raj Agrawal. The talk that preceded me, Dr. Mack, excellent overview of choroideremia. You mentioned about the fact that you're Canadian. I am Indian by heritage, and I tend to speak too fast. So I'll try and consciously be a little slow. I'm here representing Therapeutics, which is an optogenetics company. And I did notice in Dr. Mack's talk that he did not refer to the term optogenetics. So I'm going to have a chat with you after when we get some time. But I'm going to be talking to you about the clinical trial that we are currently in with optogenetics in choroideremia. So just a brief about the company before we go down the details. So Therapeutics, as I mentioned earlier, is a small biotech company based out of Berkeley, California, headquartered there. As I again mentioned, Paul Bresky is an inspirational CEO of the company. He's been in this space for a long time. And we also have Peter Francis. Dr. Peter Francis is the Chief Medical Officer, and he's been in this space also for many years. So we have, and the team is small, but everybody is motivated. We have some people here as well. We have two assets in the company. One is RTX-015, and the other one is RTX-021. The RTX-015 is the one that is currently targeted towards choroideremia and also retinitis pigmentosa, while RTX-021 is focused on Stargardt's disease as well as macular degeneration. So these assets are actually the first bioengineered optogenetic medicines which have been optimized for human use, and with the caveat that we are focusing on restoring higher visual functions in patients with no need for using goggles in these patients. So some of the other attempts require additional pair of goggles that patients have to wear after the treatment. And in our case, we don't really need that. Again, I mentioned we are led by a team of world-leading regenerative medicine and optogenetic experts. And we also have a center, as I mentioned again earlier, in Irvine, which is called the Vision Research and Assessment Institute, which is kind of unique to this particular study. All patients who are in the study are actually flown in or drive in to the center in Irvine, where we do the efficacy testing for those patients. And I'll mention a little more detail about that in the next few slides. In terms of pipeline, as I again mentioned, we have 4 different indications that we are focused on currently. So retinitis pigmentosa was the first to start off as a clinical study. This was started late last year in September, and we have, the study is ongoing at this time. While choroideremia study has started, we have dosed the first patient, and we are looking for a few patients, and I will talk about that in the next few slides again. While Stargardt's disease will start its clinical study pretty soon. So in terms of what I'm going to be speaking on, I'll go through a little bit of research in choroideremia following what Dr. Mack mentioned. I'll talk to you about what is optogenetics. I'll speak a little bit about the RTX-015, and then talk in detail about the clinical study, and lastly discuss about the Ray Center in Irvine, or what is the Vision Research and Assessment Institute. So as Dr. Mack succinctly mentioned about the various research elements that are going on in choroideremia, there are multiple attempts in different spaces, gene therapy, including optogenetics, People have tried stem cell treatments, retinal implants, which I was part of previously, and molecular photoswitch, which we'll be hearing of from Dr. Daniels after my talk. I just wanted to highlight one thing, as Dr. Mack mentioned about Corey's words about, what Dr. Bennett said. There are positives that come out, there are negatives that sometimes come out in clinical trials. Clinical trials is tough business. Some studies do well, some studies do not. And in this case, I just wanted to highlight this particular study, which got published in 2014 out of UK, Oxford. This is a phase 1/2 study in gene therapy. This was a subretinal injection where they used AAV vector encoding REP1, which showed in the phase 1/2 study that there was improvement in patients. Unfortunately, this did not translate into a phase 3 trial, which is what is required for an FDA approval so that the medications can be commercialized. So there are things that have succeeded in clinical development, and obviously some are actually more that unfortunately do not succeed. So we have to keep trying to make sure that we can make a difference to patients. So just to give a little brief about what optogenetics is, I'm just going to refresh what Dr. Mack also mentioned earlier, and just to say that in terms of how the retina functions, you have light falling on the retina, and the retina, the cells in the retina, which I'm sure everybody knows, rods and cones, they undergo a electrophysiological change in those cells, and that leads to a visual impulse. And that impulse then passes through cells like the bipolar cells and the ganglion cells in the retina, then passes on to the optic nerve and to the brain, to the visual cortex in the brain, and that's how people see. But in case with advancement in disease, in this case choroideremia, the rods and cones get damaged over time with the progression of the disease. And what happens is the light keeps falling on the retina, but there's no visual impulse that gets generated. So what can we do to target that issue? What can we do in terms of trying to make sure that we can create a retinal impulse or visual impulse that can potentially make the patients see again? And that's where optogenetics comes into play. So what optogenetics does is it recreates new rods and cones, literally new rods and cones in the visual space in the retina, which become light sensitive, meaning that when light falls on the retina, in earlier cases, the retina was not reacting to the light, but with optogenetics, those cells will now react to light and create a visual impulse that will then pass on to the optic nerve and to the brain, allowing a patient to see again. So again, it's a light-sensitive protein that we inject into the retina, into the vitreous cavity, with proteins called channel rhodopsins, which then transduce into cells. And in this case, particular case in choroideremia, we are targeting retinal ganglion cells, which then get transduced and become like a rod and cone cell, meaning if the light falls on them, they will be able to generate an impulse, and that impulse can create a visual image for the patient. In our case, the advantages of RTX-015, importantly, and in general in optogenetics, is that this works even in advanced cases of the disease. So even if the patient has significant loss in rods and cones, the retina is significantly damaged, because the inner retina, the retinal ganglion cells, are pretty intact to a significant number, we would be able to recreate a system where patients can be able to see again. Another advantage is that this is mutation agnostic, so you don't really need to focus on any of the specific mutations in this or any other disease to try and treat those patients. And also this is less invasive. We are not doing subretinal surgery, we are not taking the patient into the operating room. We just have the patient sit in the clinic on the regular examination chair and give an injection similar to the millions of injections that are given in retina clinics around the world. And also importantly, in our case, it works in ambient light, meaning in low light conditions. So you don't really need bright lights, or as I mentioned earlier, any specific additional goggles for patients to see again. So these are some of the advantages that RTX-015 has over some of the other systems that are currently being researched. Suffice it to say, we are sitting on the shoulders of giants as we work to try and make a difference in patients' lives. Optogenetics actually came into vogue, was discovered in 2003, so about 20 plus years. Visual optogenetics was defined in 2005. So again, exactly 20 years from today, previous to today. And then, a lot of work has gone in terms of taking it to where we are today. So one of the seminal years that I want to talk about is 2021. I'm sure most of you have heard of Dr. Jose Sahil from Pittsburgh. He's one of the pioneers in optogenetics, and he and his team, along with GenSight, they actually had a patient with advanced retinitis pigmentosa undergo optogenetics treatment, and that patient improved significantly. This patient had very advanced disease, significant loss in vision, but the patient showed up that there was an improvement in vision in that particular case, and that was the publication that came out in 2021. That pretty much showed that optogenetics can have a significant role to play in management of IRDs like retinitis pigmentosa and choroideremia. 2021 is also important for us at Ray Therapeutics because we actually bioengineered our proteins that year. So RTX-015 and RTX-021 were bioengineered that particular year, and that kind of led to where we are today with clinical trials ongoing. Just to highlight again, this picture here shows the retina as a cartoon, which in progression of the disease, as you go from left to right, the left is normal retina and the extreme right is where the disease has significantly progressed, where the rods and cones gets damaged. And so you don't have, no rods and cones in the retina and or very insignificant numbers. So it obviously does not give help to a patient. And that's where what I'm trying to highlight in this slide is that on the right side, we can actually use optogenetics treatment from Ray Therapeutics to try and treat patients. Obviously, gene replacement therapy is gene-focused. We also have gene editing, which is mutation-focused. And then we have neuroprotection and cell therapy that is also being targeted in terms of as a treatment for choroideremia. I'm going to take a few minutes to discuss preclinical work, not because I want to bore you with in all the details, but I just want to make sure that we all understand where we stand today compared to what the work has been done previously. So the animal model that we used in our studies was the TKO mouse model, which is definitely much better than most of the animal models that are used. Much more consistent in terms of the disease process, and also very helpful in terms of trying to define what kind of improvement a patient has, or rather a mice shows versus what a patient will have in terms of a clinical, clinical outcome. So we use an optometry system where we have an LCD monitor that is placed around the mouse, and it's a virtual rotating drum where the mouse is following some stripes that are shown to the mouse, and that's how the recording takes place about what the mouse is seeing. Obviously, the mouse cannot tell us what he or she is seeing. So this is the test that we do in terms of trying to figure out whether the patient, in this case a mouse, is able to see better in terms of visual acuity, or there is contrast sensitivity improvement, whether the mouse has improvement in mobility and stuff like that. The optomotor response test is actually very objective and it's very reproducible. So again, a very important test that we have that can potentially tell us and give us more benefit of understanding as to how it will translate into clinical study. So it focuses on light sensitivity, it looks at visual acuity, and also at contrast sensitivity. This slide basically shows what I mentioned earlier, which is that RTX-015 potentially has the benefit of working in ambient light or low light conditions. So all the way on the top, I think I don't think it shows up there. All the way on the top is where RTX-015 is, which is indicating the fact that this works in low light conditions. Other attempts require, as I mentioned earlier, additional goggles that patients have to wear, and some of the other attempts require bright light situations, so they cannot work in low light conditions. So we do have a definite, and I'm not selling Ray Therapeutics here, but I'm just saying that we do have an advantage in terms of working in low light conditions. So what did we see in these animal studies? What we saw was improvement in visual acuity. Almost 50% of those animals actually improved in visual acuity. And in terms of contrast sensitivity and motion detection, we actually see almost normal improvement in these animals. So again, like I said earlier, if you translate what we see in animal studies to humans, this is what we are going to likely see in patients. And we have an ongoing study at this time but because it's ongoing, I cannot discuss the outcomes. So what is RTX-015 again? It's an bioengineered optimized proteins that are active in low light conditions. It's got a better light sensitivity, almost 100 to 1,000 times better. It is shown to restore light sensitivity, visual acuity, contrast sensitivity, and motion detection in animal models. And importantly, it is only delivered as a single injection in the intravitreal space or in the jelly of the eye, which diffuses onto the retina and then transduces or changes the retinal ganglion cells. So let's come to this study. We call it Envision study, which is currently, as I said, ongoing. So this is a dose escalation study, which is to evaluate a single injection, intravitreal injection of RTX-015 in patients with choroideremia. We have 4 sites currently around the country. We have University of California, Irvine, which is led by Dr. Barry Cooperman, who's the chair of the department. We have Retina with his associates in Los Angeles and Beverly Hills, Dr. David Boyer, who's very well known in the retina space. He's a senior managing director there. We have the Retina Consultants of America in Houston with Dr. Kenneth Fan, who's an IID specialist himself. And then we have University of Pittsburgh with Dr. Jose Sahel, who I'm sure most of you know, a pioneer in optogenetics, and he's also department chair. So we have these 4 sites. So any of you, if you have an interest, you can obviously contact these sites in terms of discussion about the study. The study is 12 months, the initial phase, which is the active phase, so to speak, and then we have a long-term follow-up study as required by the FDA that goes on for 4 additional years, so a total of 5 years. We are looking at a total of 6 patients in choroideremia in this, but one phase 1-2 study, and obviously we will be, once we have the data from this study, we can then be planning a bigger study, a registration study, hopefully to get approval from the FDA. So what are the criteria? There are obviously multitude of criteria in the studies, as in any other study. I'm going to just highlight some of them here. So the main eligibility criteria is age has to be at 18 or over 18 years of age. We have to have a confirmed diagnosis of choroideremia. This includes genetic testing. The visual acuity that the patient has should be between 20 to 100. So when you see the chart, the top letter of the chart is what is 20 to 100. And the 28,000 is the lower end, which is close to face hand movements scenario. So these are the upper and the lower end of in terms of visual acuity. And importantly, we have an independent reading center that actually looks at the scan of the retina to define whether the patient has retinal ganglion cells or presence of a nerve fiber layer, which includes the retinal ganglion cells. And that needs to be confirmed by them before the patient gets recruited in the study. The main exclusion criteria is that if the patient has participated in any gene therapy study previously or in a cell therapy or retinal implants, unfortunately, at this time, that patient will not be considered for the study. And also, we are looking for because this is a phase 1-2 study at this time, we are looking at safety. We are also looking in terms of efficacy. We want to make sure that patient does not have any condition that can preclude us from examining the patient and knowing what kind of change the patient has. Again, importantly, safety in the patient. So what's unique about RTX-015 study or the Envision study? We do the screening and we do the safety assessments of patients at sites, meaning at those 4 sites that I mentioned earlier. But all of us know that when you go to a site, a clinical, to a clinic, they're busy. I mean, there's so many patients out there. And it requires a lot of patience and a lot of time for patients, let's say with choroideremia or with RP, to undergo testing. And so what we define in this test is we have patients who first undergo screening and safety assessment at the site. They also come to the Irvine Center to undergo baseline efficacy testing and all efficacy testing that takes place after that. And so we have the same person potentially testing the patient over and over again. So there's no variability in terms of outcomes. There is no issue with time issues or anything, because these people, these experts at the Irvine Center are very patient. And I've seen them in work, and I'm not a very patient person myself. So when I see them sitting down with the patient through the whole day, sometimes 2 days, it really is amazing how much time and energy and obviously patience they spend in terms of trying to work with the patient to see the best in terms of what the patient sees and stuff. So really something that is unique in terms of this study. So what we have is we have about 5 visits during the first year of the study. So all these 5 visits will happen in Irvine. If you live outside California or you live up north or something of that nature, we'll fly you in to the Irvine Center to undergo the testing. And obviously you stay there, and if it requires 2 days, and then the testing can be done there. Plus, as I mentioned, we have a long-term follow-up study as part of this particular Envision study. So there'll be 6 additional visits over the next 4 years. So again, RTX-015 choroideremia study has been initiated. It's an open label. So obviously there's no placebo. There is no, kind of a safety side of things at this time. We're looking at safety in the patients themselves. It's a dose escalation study. We only have 2 cohorts, so mid-dose and higher dose. And we are looking for 3 patients each in each of these cohorts. And like I said, 1 patient has already been dosed. We already have an ongoing RP study that is currently ongoing as well, and again at these sites. And so far, I can just mention the fact that we have seen excellent safety in those patients so far, so which is definitely hopeful for all of us, because one of the challenges that gene therapy studies have seen previously is the issue of safety. Some of the patients end up with inflammation and things of that nature, and in our study, we've not seen anything of that so far, which is great. Lastly, the Vision Research and Assessment Institute, just to highlight the fact, and importantly, this is led by world-renowned low vision experts who have expertise in endpoint validation as well. And the reason why I mentioned that is because IRD space, including choroideremia, we have an issue with endpoints. When we look at the regular FDA-defined endpoints, 3 lines of improvement, sometimes it does not suffice in terms of improvement in patients with diseases like choroideremia. So we are trying to work out avenues to see whether we can come up with new endpoints that can potentially convince FDA and other regulatory agencies so that they can accept that as a particular endpoint. So we're working on that at that particular center. We're also looking at, one of the things that we also have an issue with is different days. Patients tend to have different outcomes. If you come one day and you come 3 months later, the outcome that you may give in terms of what you see and what you experience may be different. So there is test, retest variability that happens in patients and also from the person who's testing you. So what we're trying to do is we're trying to define whether if a single person does the test, would that test, retest variability be reduced? So that's also a study. Actually, that's a study ongoing at Ray in Irvine. So if you're interested in getting involved, speak to Reem. Reem, if you can wave your hand. So she is going to be at the booth outside. So if you want to talk about the study or anything in relation to what we do, you can speak to her or any of my other 2 colleagues as well. I think that brings me to the end. Again, I'm just giving the phone number and the email ID here for anybody who's interested in reaching out to Ray Therapeutics. Again, we are the sponsor for the study, so we don't talk to you guys if you're a potential patient. But Reem is firewalled, so you can talk to her and you can discuss the details with her. So thank you again, and great to be here and speaking to you all. Thanks again. Any questions? Why don't you speak up? I can repeat the question for you then. Thank you. I think those are really important questions. Number 1, the question was, how long is this effect going to last once we give the injection to the patient? And we have done studies on this to figure out what is the lifetime of the treatment. And what we believe at this time, based on what we've seen in animal data, is that this will be one and done situation. So you give an injection and that'll be it for a lifetime of treatment. Again, other studies have shown that sometimes gene therapy effect does reduce over time. So we don't know unless we follow the patients over time, and that's what we'll be doing. But at this point, we believe that will be a one and done situation, so 1 injection. And the second question was about why do we have the patients come in for so long? So that's a good question. And the question again is just to repeat, why do we have 5 years or a total follow-up versus 10 years that other studies have required previously? So I think this is based off on the FDA requirement. FDA requires at this point of time for patients to be followed up for 5 years and not 10. We will be following up the patients even beyond that. So obviously, we want to see what the long-term effect of the treatment would be, but defined study criteria requires us to follow for a total of 5 years. Thank you.
So thank you very much. So we can let the next panelist speak. I think for the moment we'll hold off on questions. Ray Therapeutics is out in the foyer area. Also, they'll be taking questions at any time. And
Thank you.
Thank you
Thanks so
very
much.
much.